This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 337, recorded on July 24th, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. We have summer here.
Yeah. Is it 90 degrees?
100.
Oof.
Wow. That's amazing. Here it is 30 C, which is pretty warm. Also joining us from North Michigan, Michelle Swanson.
That's right. Good to see you all. Yeah, we call it up north.
Up north. Is it mild weather up north? It's probably cool, right?
Yeah, it's like in the mid 70s today, which is lovely.
That isn't a Youper thing, right?
No, I'm not over the bridge.
Okay.
I'm still below the bridge, a troll.
That voice you heard, since you can't see us, you can only hear us, is our guest today from Tacoma, Washington, Mark O. Martin. Welcome back.
It's always a pleasure to be here. By the way, Vincent, it is 23 degrees C here.
Yeah, mild. Very mild. Does it ever get really hot up there?
Oh, yeah.
It does?
That's not good. We don't like that.
Well, it's weather, you know. What can you do?
Mm-hmm.
Wait.
Wait. Jump in the water.
Burn fewer fossil fuels? I don't know.
Ooh, can't say that.
Oops.
Well, you know, as we'll hear today, anthropogenic activities really, it's it that messes everything up. You know, we can't blame anything else except ourselves, but the alternative is, well, humans could behave better, but they don't because they're driven by power and money, unfortunately. Nevertheless, this is microbiology, and we're going to talk about two interesting stories, which are completely opposite, although they could be linked, as you will see. And Michael's going to tell us what happened many thousands of years ago with infectious diseases. This is a very cool story, Michael.
This particular paper is entitled The Spatiotemporal Distribution of Human Pathogens in Ancient Eurasia. And it's an open access article, which is unusual for Nature, and it was published in Nature on July 9th, 2025. And it's the result from a massively collaborative effort led by Martin Sikora and Eske Villerslev, along with 23 other authors. And since it's a snippet, if I name them all, I'll be done. And they're from many different institutions across our globe. This is truly a global paper. The second sentence of their manuscript captured my imagination and literally sent a chill across my body in this hot July summer. Until 1850, at least a quarter of all children died before the age of 1, and around another quarter before turning 15.
Oof.
Infectious diseases are estimated to have been responsible for more than half of these deaths. So why the chill? Well, as our society enters into an era where our anti-infectives are no longer effective, what are we going to do if we have to go back to the bad old days when we don't have antimicrobials that have transformed our lives?
Well, Michael, here's a novel concept. Maybe we could vaccinate.
Oh, no. You said the V word.
Vaccinate and fund biomedical research to advance new therapies.
And I mean, Michael, the reason why this is no longer the case, right, that a lot of kids die of infectious diseases because of antimicrobials, antivirals, and vaccines and public health measures, right?
Yep.
Absolutely.
And that's what we need to keep doing. It's a proven approach.
So we're going to draw some parallels from our ancestors here. And what this group of workers did is they wanted to create an archaeogenetic-based map of human pathogens across 37,000 years of Eurasian history. So we're looking in Europe and Asia. In other words, what was afflicting our human ancestors all those many years ago at the time of their death? We don't know if these microbes killed them. We simply know they died with them. Now, the field of archaeogenetics is a recently evolving and truly interdisciplinary dependent area of research where the intent of the investigators is to study ancient DNA, to effectively ask questions through an interdisciplinary lens that are addressing questions brought to them by a group of interdisciplinary set of investigators. Now, the questions might concern whether there might be a genetic relationship concerning the migration of a pathogen before human air travel, or even the evolutionary past of past populations. Now, not only did I find this article intriguing, Carl Zimmer, in his piece in the New York Times, and we'll drop that into the show notes, but unfortunately it's behind a paywall. He introduced us to the goals of the authors by reminding us of the famous chronicled account of the plague of 430 BC that ravaged Athens, Greece. And many of our prospective freshmen are probably reading Thucydides, where he described the gory details of this infection in his history of the Peloponnesian War. And quoting from Zimmer's article, the inward parts, such as the throat or tongue, becoming bloody and emitting an unnatural and fetid breath. And Zimmer points out we still don't know definitely what the microbe was, but the best guess is, of course, it was an enteric pathogen, Salmonella typhi. And so what all of this involves is analyzing genetic material extracted from archaeological remains like bones and teeth or other well-preserved tissues to gain insight into what people died with all those years ago. Now, this interdisciplinary field combines techniques from archaeology, genetics, and other related disciplines. Now, here, the authors described results from a shotgun sequencing approach of nucleic acid that they extracted from ancient human remains. And they looked at 1,313 samples to give you an idea of the scale and scope of the human remains that they evaluated, looking for evidence of infections at the time the body was buried or preserved.
And if I could interject, Michael, this covered a span of 37,000 years.
Absolutely, Michelle.
It's a really ambitious project.
So their question was really straightforward enough. What microbial nucleic acid could we uncover at the time the bone, if you will, went into the ground? So what did they find? Up front, they offer, and they were very careful in couching their analysis. They didn't overgild the lily. They offer that their analysis was biased. And I don't know if that's the right or proper term, probably narrowed. But what they did is they selected a set of 136 bacterial and protozoan genera, which accounts for 11,500 species, give or take, containing human pathogens. As well as for Vincent, they looked at 1,356 viral genera, which encompasses 259,000 species in total. For further,
they did it just for me.
They did it just for you. Well, you're the Earth's virology professor, Vincent. So they uncovered a widespread presence of ancient bacterial and viral and even parasitic DNA, tallying up 5,486 individual hits against 492 species from 136 genera of microbes. Crucially, 3,384 of these hits involved human pathogens, many of which have never been identified in ancient human remains before. Now, this is a bioinformatic rich paper, but it's really not that hard to get into. You just have to look at their 3 figures. And since it's open access, the first figure provides for you a snapshot and a temporal window of where their samples came from. Their second figure describes what they discovered, and the third figure is from which era the microbes were from. So you really get a sense of what they were uncovering. And as you might expect, they commented on this as well as the rate of read mapping varied by orders of magnitude between species. Now, the high hitters were things like Mycobacterium leprae. And that makes sense. It's a disease that made the Bible. So ancient humans knew about leprosy because they describe it in, one of the oldest texts that we have. The lower hitters, bordering on the limit of noise, were able to witness a louse-borne pathogen, Borrelia recurrensis, which is responsible for relapsing fever, and it's a spirochete. They also found things like soil microbes like Clostridium botulinum, and they were found in similar rates from both bones and teeth, suggesting that these folks actually had it in them. Because remember, you're recovering the nucleic acid from effectively inside the tooth or inside the bone, because the surface is not that accommodating for recovering ancient DNA.
And the other challenge that they point out is that there could also be contamination from the burial process itself. So they had to screen out what they called the necrobiome.
The dead body effect.
Yeah. Whatever burial practices were common in that period, in that area.
And one of the big takeaways from the results, aside from the fact of answering who was resident at the time of the person's death, was their observation that zoonotic pathogens, those microbes that we acquire upon being close proximity to animals, only began to appear in the nucleic acid record starting about 6,500 years ago, and they began to peak about 5,000 years ago. Now, granted, they had significantly fewer samples from earlier time frames, but they offered that the early farmers of the ancient time weren't succumbing to the diseases that the folks from 6,000 years ago. So what's so special about that time frame between 6,500 BC and 5,000 years ago? Well, coincides almost perfectly with the widespread domestication of livestock. So by using microbes found at the time of death, this study provided to us direct evidence that this shift in human lifestyle from our previous way of living, hunter-gathering, to an agricultural and pastoralist societies led to a significant increase in infectious disease burden within the human race. Again, we were crowded into villages to effectively protect ourselves. Now, the older remains were from our hunter-gathering ancestors, and their bones and teeth harbored a host of pathogens from hepatitis B to the stomach-dwelling microbe Helicobacter, where longtime listeners of TWiM might remember we covered its meandering journey from the Fertile Crescent through Asia and across the land bridge into North and South America by just looking at genetic drift in the Helicobacter genome. Now, let's not tell ICE that microbes actually smuggled their way into North America, but the early remains did not have traces of the big killers of recent history, like Yersinia pestis. They only saw its rise in the ancient remains starting about 6,000 years ago. And remarkably, they failed to find traces of pestis, the plague microbe, in the remains of early farmers. Rather, their study offered that this nasty microbe's origin story pointed to the nomadic tribes of Russia and Asia, where these tribes were rearing vast numbers of livestock like cattle and horses, which would likely generate a better environment for microbes that could transfer the bacteria from the animal, these are zoonoses, to the human host. And these, of course, are fleas and lice. They could not say for sure, but any student of One Health will appreciate that the central theme of the One Health initiative, namely, is to offer us all a data-driven understanding that human health, animal health, and environmental health are interconnected and interdependent. However, this may be also, the authors offer, a function of differences in the immune status of farmers to nomadic tribes. And it didn't start there. They also observed that the spread of these pathogens surged during subsequent millennia, aligning again with the sedentary lifestyle migrations from the Eurasian steppe all the way into Central and then Northern Europe itself. This really underscores how deeply intertwined our history is with the microbial world and how our societal changes can profoundly impact our susceptibility to disease. And in fact, the intensity of some of these epidemics were so intense that they could be eyewitnesses to the selection pressure that then resulted in a spike of mutations in the immune system of the nomads that simply boosted their ability or enabled their immune system through mutation to thrive. Or it could be something completely unrelated to the CCR5 mutation that we see in plague survivors, but rather could be something simpler, like they simply had a greater abundance of protein secondary to the meat supplied by the livestock that they were rearing in these gigantic herds to enhance the resistance to the pathogens. And then there's body lice. Now, Borrelia recurrensis, the relapsing fever of the Neolithic farmer that they found in 10,000 BC in Scandinavia. And then, of course, Mycobacterium leprae. They attribute leprosy spread was associated with red squirrels and humans. Of course, the fur industry, as you're skinning animals, you, of course, are exposed to any of the insects that are affiliated with the furs, and the tanning process would make you at risk. Or should you nick yourself during the skinning process and the tanning process, you may inoculate yourself. And all of this occurred during the late Iron Age and the Viking Age. And then, of course, let's not forget the diseases of the desiratory tract, namely syphilis. They saw syphilis pop up between 500 and 600 before our current epoch. Again, here, this is another fantastic example of using ancient DNA to understand evolutionary history of human pathogen interactions. However, with any groundbreaking study of this magnitude, there's always some things to consider, some limitations that are important to acknowledge. And the authors, again, are quite upfront about this, stating while ancient shotgun metagenomic data offers direct evidence, there are challenges. For instance, they had struggles with eukaryotic pathogens like some parasites. They can be particularly daunting due to sequence contamination that Michelle mentioned earlier and the complexities of distinguishing true ancient DNA from environmental contaminants. This will always be the case, but this still does not diminish the significance of their findings. They've done an incredible job with the data available. We have to remember that we're dealing with degraded and fragmented genetic material from teeth and bones. The presence of DNA doesn't always equate to active infection or severe disease, and inferring pathogenicity from ancient DNA alone can be tricky because we can't even do it today. And we simply know that the individuals supplying the sample also harbored the microbe at their time of death, which likely explains leprosy because it's a long-term chronic disease. Also, the presentation of microbial DNA varies depending upon environmental conditions, type of pathogen, and the fossil records of the pathogens, of course, is inherently biased. Some pathogens might simply not preserve well over thousands of years. They don't have a predilection for bone. They don't have a predilection for teeth, potentially leading to an underestimation of their historical presence. So while the broad strokes of their conclusions regarding the impact of lifestyle changes are robust, the fine-grained details of every single detected pathogen requires careful interpretation and further study. Despite these inherent challenges, the significance of this work cannot be overstated. It provides a unique window into our past, offering direct molecular evidence for the deep history of human infectious diseases and the coevolutionary dance between us and our microbial companions. Again, their data serve to reinforce the central theme of the One Health initiative, which is to offer us all a data-driven understanding that human health, animal health, and environmental health are interconnected and interdependent. We need to continuously remind some of our so narrowly focused colleagues the importance to recognize the interconnection between people, animals, plants, and their shared environment. We cannot simply legislate it away or say we need more coverage in the curriculum for AI. For the data here serve to help us understand the long-term impact of human transitions like agriculture and migration on our health landscape. Today, we might imagine this as global climate change, for it has already impacted our entertainment industry by providing us amusement through the development of a fictionalized zombie-generating fungus, like the version of the real-world Ophiocordyceps unilateralis, also known as the cordyceps fungus, popularized in the HBO Max series, The Last of Us. So in closing, this kind of archaeogenetic work is vital for building a more complete picture of human health and disease dynamics over millennia, and most importantly, into our future. I believe that the data provided by this multinational group will reinforce the idea that pathogens have been a constant selective pressure through our evolutionary journey and will continue to do so in spite of what we legislate. So there you have it, a deep dive into ancient pathogens and human history. It serves as a reminder that even way back when microbes were running the show, just like they're running the show today.
I would love to see this kind of study in the Americas.
Yes.
For a number of reasons. First of all, that many of the infectious diseases in the Americas were brought over from Europe, right? So we could see that happening, presumably. But also there must have been already infectious diseases present, and we don't know much about that at all. Nobody really talks about it. They just talk about the conquistadors coming to the U.S. and decimating the population with their infectious agents and measles, but they must have had their own. Why didn't the conquistadors get infected and back, you know? So I'd like to see that. Also, there's a statement here in the conclusion. They say zoonoses account for more than 60% of newly emerging infectious diseases. That's not correct. These are zoonotic origin, but once they become human viruses, they're not zoonoses any longer, okay?
Same with bacteria. They're no longer zoonoses.
But the One Health concept still holds. They originate from the animal.
Of course. Most of our diseases originated from either our ancestors or Neanderthals or Denisovans or Australopithecines or non-human animals. There's no question about that. All of them, they all arose from some other source. But today, human influenza is not a zoonotic infection. It goes from human to human. Okay. When we have a new pandemic, that's a zoonotic spillover, but then it becomes a human virus. So that's just a minor thing there. But people often confuse this because they confuse zoonoses with zoonotic origin. That's all.
One really neat example of the One Health and the zoonoses was they mentioned that the agent that causes leprosy, M. leprae, can grow not only in humans, but also red squirrels. And they detected the rise in human sequences, of leprosy in the teeth, at the same time that the fur trade from Scandinavia, especially squirrel fur, was rising. So they were able to show that these were coincidence, which is a great example of One Health.
And the rise of Yersinia enterocolitica, which is principally an intestinal disorder associated with eating poorly preserved meat or undercooked meat. They also saw the rise of it during the time the large agrarian societies were raising livestock for protein. So it's really
so the question
fascinating.
is, folks, would you rather remain a hunter-gatherer and have fewer infectious diseases, or do you want to pay the price? You want to pay the price, huh? Yeah, because if you don't get an infectious disease, a lion's going to eat you anyway. So
And pay the price. Do you want to elaborate on that?
pay the price for infectious diseases. Oh, so we have a lot of infectious diseases now, but we have a society that arguably is good, right? Technology is amazing, medical advances, public health, or you could live in a cave and be afraid.
Right.
That's the trade-off.
So we need to embrace public health.
But the one thing is, if our children of 37,000 years in the future ask the question what we're dying from, and what we're dying from is principally cancers. They're not going to see that unless the cancer cell somehow manages to end up in your tooth or in your bone.
That's right. I don't think they had many cancers because people died in their 30s, you know?
Right.
So there were childhood cancers, I'm sure, but
they were died.
No cancers
died quickly.
And no dementias, very few dementias as well, right? And so life is a trade-off. That's it.
Yes.
Thank you, Michael. Okay, now our guest, our TWiM adjacent, Mark Martin, is going to talk about plasmids, which I love. I got trained on plasmids.
I think we all did. I
just use them as tools. I use them as tools, and I'm going to tell you some stories that you'll like. But go ahead, Mark.
Oh yeah, no. I'm a big plasmid fan for all kinds of reasons, and I want to say to you and to listeners, this is a very interesting choice for me. I adore plasmids, but I'm not super comfortable with mathematical analyses or computer science, so I'm batting about 50 here. So I'm looking at all of you to kind of keep me honest about stuff as I hit the high points. But I before I start, I want to ask everybody. I'm going to start with Michelle because you're the furthest to my left there. I don't mean that politically. What do you think of when I say the word plasmid?
Genetic engineering.
Yeah, I'm not trying to trip anything up. I'm curious. And Vincent, how about you?
Oh, tools for cloning. Yeah.
Michael?
Something the microbe can live without.
Well, that's interesting in all ways. I was very fortunate, and I'll keep this kind of stuff short, but I want to say that when I was in graduate school, I worked with an organism that didn't have a genetic system, so we used a lot of unusual plasmids for things. And I quickly learned that they were in business for themselves. And specifically,
the plasmids were.
Yep.
Oh, yes.
Yeah. So would you say plasmids are parasites?
That's a wonderful question. I love the idea of it, but I find that every time I quote Dawkins, the selfish gene, people get angry with me. So
I think of
them as lost luggage, lost nucleic acid luggage that you think you might be able to use, but you don't really know why you're carrying it around.
So Mark, maybe you should define a plasmid before we go any further.
Oh, I will. It's an extra chromosomal replicative agent. Okay.
How about in English?
So it is a piece of DNA that can replicate apart from the chromosome. And I want to note to listeners in particular, so many of us who are microbiologists have been co-opted by the idea of them being tools, and wonderful things have happened. In addition, we think about plasmids that carry as many as 5 or 6 different antibiotic resistance genes. So we focus on that. And we're not thinking about, quote, the natural history of the plasmid. So what I started to say is that when I was in graduate school and I worked with what was then Rhizobium, but is now Cynorhizobium mellilodi, we found out it had two enormous plasmids. And we very creatively called them megaplasmids because that's how we are. Now, one of those megaplasmids carried a couple of symbiosis-associated genes. Rhizobium forms a symbiosis with the roots of alfalfa to form nitrogen-fixing root nodules. The other ginormous plasmid had at least one gene for thiamine biosynthesis. I have to admit shamefully that I didn't dig in to see what current annotations look like, but I will come back to that. These plasmids were 1.3 and 1.4 megabases in size, and the mellilodi genome itself was 4 megabases.
Wow.
So is it a giant plasmid or a small chromosome?
Or a parasite.
And we'll come back to that. Are they selfish DNA elements that pick up hitchhikers? Are they the architects of microbial evolution, or both? And I often will ask students, is it A or B? And then they'll pick one and I'll say, it's both actually. And so today's article, Universal Rules Govern Plasmid Copy Number, was published on the 2nd of July in Nature Communications by Ramiro Martinez de Quinto, Lanza Gama, and Rodriguez Beltran from, please forgive me, the Hospital Universitario Ramon y Cajal at the Universidad Autónoma de Madrid and CyberInfect in Madrid, Spain. Yes, a number of different people in Spain. Now, I also want to say to all the listeners that this fascinating paper was published back-to-back with another paper, Scaling Laws of Bacterial and Archaeal Plasmids by Matasetti, Shaiti, Wilson, San, Baig, Zhou, Liu, and Yu from Duke University, Rutgers, and Stanford. Now, I hope that I can make that article available in the show notes as well, because it has many congruent ideas that I'll touch on and some follow-ups.
And if I could amplify that,
Please.
I was able to get in touch with the first author, and she pointed out that when she went to the International Symposium on Plasmid Biology in Hamamatsu, Japan, she had the good fortune to meet one of the investigators who was independently discovering the biology that you'll describe here. And they just had a wonderful conversation and synergy, and they ended up agreeing to submit their papers and publish these complementary studies back to back in Nature. So she said it's an example that science is at its best when it's collaborative and fueled by curiosity, trust, and a shared purpose.
That's so well put, Michelle, and thank you. I always look forward to you talking to the authors. But I wanted to say that as I went through this paper, I kept writing little objections that the other paper solved.
Oh, good.
And so what's interesting about that is that they didn't write these papers together. They came at it in different directions. Because these papers have a lot of acronyms, I just want to run through the four that get used over and over again. PCN is plasmid copy number. PTU, that I'd never heard of, is plasmid taxonomic units. LCP is a low copy number plasmid. That means between 1 and 2. And HCP is a high copy number plasmid of over 10. Folks, it's important to remember that most of the plasmids that we work with in molecular biology and microbiology, especially when we do cloning, are usually at hundreds of copies per cell. You can sometimes change that with things like phosmids that are kept at 1 copy.
Okay. Does anyone remember the first plasmid that was made widely available to non-microbiology researchers?
I'm guessing PBR 322?
No, there was one before that. It came out of California.
PACYC?
PACYC, which low copy number, because if you had toxic genes that you were trying to put in, you needed to use a low copy. I tried using it. Man, it was hard to work with.
Yeah.
It was.
PBR is high copy, which then followed, and that most people get along with PBR. But nowadays, nobody uses PBR anymore, right? I don't even know what people need. People don't clone anymore. They don't have to.
Nope.
They just sequence and produce it.
I do. and it's sometimes a problem.
But you're just a dinosaur, Mark.
Oh.
Let's get on to the story.
Thank you. Now, there are several strategies for maintaining plasmids during cell division, because if you lose a plasmid during cell division, that's not in the plasmid's best interest. And yes, I'm inferring intentionality.
Ooh.
Now, one of these is based.
I would say it's not a good evolutionary strategy.
Yeah, it's a dead end. And so what you find is that one strategy is based wholly on high copy number. Now, if you have a couple hundred colonies per cell, stochastically, you're almost certainly going to get some in the progeny cells. But many plasmids, including the very famous F1, they're one copy per cell. And how do you do that? And there are several different techniques. The one that I enjoy teaching to students is the toxin antidote system, just to ensure that every offspring cell, every progeny cell has to have the plasmid.
But Mark, if you have one copy per cell, the two daughter cells, one of them is going to lose, right?
Yeah. So you have to make sure that the replication and the partitioning take place in a coordinated fashion.
Right.
So getting to the paper, enough about me. The authors, I love plasmids. I worked with so many weird plasmids. And by the way, that's how I got to know Esther Letterberg at Stanford, because she was head of the plasmid repository.
Yeah, she tried to control the naming of plasmids.
Well, they did get a little strange after a while.
I got letters from her saying, you can't use that name.
So the authors of this first paper looked through DNA sequence data and they calculated plasmid copy number. And as you read the paper, it's PCN here and PCN there, from 6,327, quote, phylogenetically diverse plasmid. I'll come back to that. They found out that PCN is highly variable from plasmid to plasmid, but each plasmid maintains a copy number that's stable and characteristic of that plasmid. In my mind, I'm thinking because they're entities.
And these are across many different genera, right?
Well, we're going to come back to that right now, in fact. So they found a conserved negative relationship between plasmid size and plasmid copy number, independent of the bacterium hosting it, and what's called a universal scaling law. But let's go to the details. They did 9 different genera, but if you look carefully, they're mostly looking at pseudomonads and bacilli. 95 species, but their focus is mostly on things of medical or biotechnological importance. They identified 139 plasmid taxonomic units, 238 different replication types. This is based on what's called incompatibility. And I love this, 2,200 sequence-based clusters. This just means that they have a lot of different plasmids to look at.
And again, if I could interject,
please.
The first author shares that they spent months downloading large amounts of sequencing reads, often running analyses on 6 or 7 computers simultaneously because they had so much data that they were processing.
And one thing that they talk about, and since you spoke to the first author, presumably you heard this, is they had to be very cautious about the quality of the data because there's a lot of stuff up there that isn't quite polished enough for what they want to do. So what they're interested in is they're determining the plasmid copy number, and they do that with the coverage of the plasmid in terms of amount of DNA versus the coverage of the genome. And thus they have 6,327 high-quality plasmids and genomes to look at, and the copy number shown in figure 1.
So, Mark, you can get copy number from the read number of the sequencing?
It can be really complicated, and they're very cautious. I'm glad to hear you say that, Vincent, because, for example, you don't know about the culture conditions.
Yeah.
If you got them early or late, and there's a lot of heterogeneity within a culture, of course. And so when you look at figure 1, what you're going to notice is there's a bimodal distribution.
But if I could get back to Vincent's question.
Please.
What they used essentially as a denominator was the chromosomal copy number.
Yeah.
So then it was plasmid copy number or sequence read load versus chromosomal load.
And the chromosomal copy number is a known quantity for these bacteria, correct?
This brings us to this.
Well, the chromosomal number is a function of the growth phase and the nutritional status of the community that's effectively developing.
Yeah.
And that is reflective because when you're in exponential growth, that is when folks believe, if they've never proven, that the population is synchronous, that you effectively have the same number of replicating forks in every chromosome, and the plasmids are tuned so that every daughter will get an equal distribution of plasmids. And it's really hard to demonstrate until we get to single cells.
So this makes me so happy when I'm laughing, because these are all questions that I had.
I'm sure they're on your paper.
Well, yeah. And so for example, I mean, if you look, just to be clear, if you look at figure 1, I just called it up to have a look. They have some nice illustrations of the number of strains, number of plasmids, replicon types. One more time, replicon types are usually based on what's called an incompatibility group. That is to say, plasmids that kind of duke it out for materials in the cell. And if you go to figure 2, there's the bimodal distribution that we talked about. It looks like there's groups with many copies and groups with few. And that's really interesting to see. So again, the plasmid copy number is quite broad across the data set. And they say, and the microbial gods bless them, this could be due to overrepresentation among the enterobacterial relatives. And it wouldn't be at all a surprise, and they're very upfront about it because that's where their focus was. Okay? They basically found that the high copy number plasmids were based on the type of replicons, and we were talking about PB322, like coli-1, and rolling circle mechanisms of replication. And that makes sense. They make tons and tons and tons of plasmids. Now, the low copy number plasmids, however, were based on some of these incompatibility group F plasmids, similar to the RP4 derivatives I used in graduate school that were, I think, at 4 or 5 copies per cell. What's interesting to me is plasmid mobility, that is to say, the ability to conjugally transfer from one cell to another, seems to be more common among the low copy number plasmids.
Which makes sense.
So,
they can't afford to be low copy if they don't have a very precise mechanism for transferring the plasmid.
Yeah, right.
Yeah, so we cheated with that in graduate school because we put the mobility site from RP4 into PBR322. And by the way, Vincent, we would get multiple hits on transposons doing that, and that's because there were so many copies of the darn thing, just as we said. We should have used PACYC. So the other thing I wanted to say is that, and this is really important, the plasmid copy number appears to be independent of genes, host, or co-resident plasmids. And that suggests that copy number is controlled by the plasmid. And, you read that and think, well, of course. But there are many cases of extra chromosomal entities that take advantage of host mechanisms. So that's kind of interesting.
Mark, do any plasmids encode their own DNA replication systems?
I have never seen one that does.
Yeah. Okay.
I've never seen one that does.
Okay. So they really are parasites.
Yeah.
And it's interesting because then you think, well, what's the cost? What's the benefit? And we'll come to that. So, there appears to be more variability in plasmid copy number among the high copy number plasmids. Now, when we say variability, I want to be clear, when you dig down the data, they're not talking about 2 copies versus 1,000. They're talking about 1 copy and 10. So that is variability. And then they did something that I was very impressed by. They made plasmids that had 2 origins of replication, essentially, from different plasmids to see if the replicon would duke it out. In other words, was there more variability in these plasmid hybrids? Was one type of system dominant over the other? And amazingly, you could have knocked me over with a microbial feather. Plasmid copy number is in these hybrids is controlled more by the low copy number plasmid replicons. And that really surprised me. Then this is the weird part. And if you go look at figure 5, you can see the diagrams. Plasmid size and plasmid copy number are inversely correlated in a scaling law fashion across orders of magnitude. And again, let me call it up here. When you look at...
Yeah, and tell us what scaling law means. Tell the general audience.
Absolutely. Scaling law refers to something that works across a broad distance. For example, size versus metabolic rate is a good example. With plants, again, size and an amount of seeds that are being set.
So they're directly correlating.
Yes. And so when you look, for example, at figure 5A, you're looking at, I'm looking at Escherichia, Klebsiella, Enterobacter, and a nice straight line goes through all of them.
Diagonal.
And they're very careful. Yeah.
Yeah.
And they basically say, and this made me so uncomfortable.
So Mark, just say it's a diagonal when you have copy number versus plasmid size, right?
Yes. A 1% increase in size is associated by a 0.65% decrease in plasmid copy number.
On average.
And on average. And this to me didn't make sense because of the megaplasmids I worked with in graduate school, right? Anyway, I was amazed by this, and I have more to say about that. That's really cool. So the plasmid DNA load appears to be conserved relative to genome size. And this is the part you got to look at the data, and it's on average, all plasmids appear to account for the same percentage of the host genome.
It's amazing.
So if you have 4 or 5 different types of plasmids, they add up a little bit. So I'm thinking about Rhodobacter that appears not to have a chromosome, but a whole bunch of plasmids, right? So they're saying 2.5%.
They're not saying it. That's what the data show.
Yes.
Thank you. Now, I want to be clear, and I was so pleased that they were so careful about this. They were straight up about possible limitations to this work. For example, the taxa that they chose, even though they tried to be quite broad, there were a bunch of issues with clinical sources, obviously, and things involved with this. There might have been technical issues having to do with, at the time DNA was harvested, and we've talked about that. And the fact that they're only looking at circular plasmids when, of course, there are also linear plasmids in bacteria. So that was a remarkable paper, and I liked it. I was pleased with it. I recognized the amount of work. And then I looked at the adjoining paper. Now, the adjoining paper is by, as I mentioned, Madamsetti et al. And they used a program, a computational model. And it's pseudo-alignment and probabilistic iterative reassignment. And they used the NCBI reference sequences and the sequencing read archives. And they looked at 4,644 bacterial and archaeal genomes. And they harvested out 12,000 plasmids. And in the interest of time, I'll keep this nice and short. There are 3 things that they come back with is this. Again, this inverse power law correlation between plasmid copy number and plasmid size, that diagonal that Vincent and I were talking about. Number 2, that there's a positive linear correlation between the protein coding genes and plasmid size. And it's easy to say, well, of course, but sometimes we have DNA of no known function, right? And then 3, a positive correlation between metabolic genes per plasmid and plasmid size, especially in large plasmids. This is one of the things that's a problem when we teach microbiology is we pretty much look at plasmids either 1, tools for molecular biology, or 2, as reservoirs of antibiotic resistance. I mentioned earlier, we talked about this before, they found that many of the large plasmids actually had a plasmid copy number of under 1. And you think, how could that be? And they posit polyploidy of the host chromosomes. And this at the end brings me to my very favorite comment.
Sorry, Mark.
Go ahead.
Polyploidy explains a copy number of less than one because there's so much host DNA and according to this relationship, there has to be a small amount of plasmid, right?
We always presume there's only one bacterial chromosome present, when of course that might be true in clinical isolates, but it's not true in a lot of environmental ones. And so their take-home lesson, I want to quote it, I'm so pleased about this, as plasmids increase in length, they converge toward chromosomal in copy number and functional content, and this challenges traditional distinctions between plasmids and chromosomes. And that brings us back to the issue of the megaplasmids I mentioned before. So I was really excited to read both those papers together, though I was really only supposed to present one. If anyone decides to read the papers for themselves, and I hope you do, they dig into the probabilistic and computer science weeds, but it's so nice to see two different approaches that gave the same kind of result.
So, Mark, I'm trying to understand what is fixing the amount of DNA such that you have this relationship between copy number and plasmid size. Well, is that because the total amount of DNA in a bacterial cell has to be fixed?
I'm very nervous about it.
I think if you think about it, Vincent, it probably has to do with how much of the nucleotide pools can you produce given your resource budget, because the function of the cell is to divide, and they have to make lipid, they have to make protein, they have to make other things. And nucleic acid, especially in the polyploidies, and the good example of polyploidy is you look at the cell division time of Clostridium perfringens. It divides in 9 minutes, and the replication time of the DNA polymerase is pretty much the same across many different genera. You basically have the same speed. And the only way you can divide in 9 minutes is if you have many, many, many replicating forks, so that at any moment in time, you may not have a complete chromosome, but you got all the parts. You just may not have put them all together. And I think it's a resource limitation. And the plasmid, this scaling factor, really is getting to the nutritional aspects. And remember, bacteria are tremendous because of their surface to mass ratio. They have tremendous surface area that enabled them to bring in substrate to make more bacteria. But they're limited by the efficiency of their enzymology and the compartment size, because all of this chemistry is aqueous. And remember the universal definition that all of Mark's students get right on his test. They have no organelles. They have no little cubicles to make stuff. It's a vat of stuff that they're doing all these syntheses in.
Is that right, Mark? Organelles are the key to complex life?
Well, I think that you need to, and there are things like organelles in bacteria and archaea,
and they chlorophyll,
and they serve the same function. It's like a bento box. That's what life is, right? And what I want to say is this idea of resource allocation is something they did talk about in both papers. And what I would add to this is that when we talk about polyploidy in bacteria, we focus on things like Eupulopicium, where you have thousands and thousands of genomes. And I used to tell Elio that was all about serving the cytoplasm because of distance and diffusion rates, because of the huge amount of cytoplasm in that particular cell. But we don't think about what Michael just said, is there are many cases of bacteria that grow extremely quickly But yet the amount of time it's estimated for them to replicate their DNA is much longer. So they have multiple rounds of replication taking place on the same replicon. So with that in mind, there's a lot of polyploidy that we don't think of. To add to that, Vincent, we don't know how the organisms from which the DNA was isolated, how they were treated necessarily. And I don't know how a cell, an E. coli cell in my gut, versus in a 2059 tube shaking at 37 degrees. I don't know how different they are in terms of their replicative speed or structure.
Yeah, and what you're pointing out is that this was kind of akin to an observational study rather than one that you design with all the controls. Yeah. But that is one of the powers of doing a purely computational study. You get a vast amount of data, see if there are any patterns, and they clearly identified some very convincing patterns, some rules. But they also were very careful to point out what the potential limitations are and said this is now a start to look at these questions in more detail. And they includes our first author, Paula Romero Martinez. She's a PhD student with Dr. Geronimo Rodriguez Beltran, and she arrived with great training, a degree in biology, a master's in biotechnology, and as she's been working toward her PhD, she also completed an additional master's in bioinformatics and computational genomics. And this study, she says, is one of the first of the Rodriguez Beltran lab that is purely bioinformatic. So good for you. She got interested in science as a kid. She always loved pretending to be a science, mixing things, inventing experiments, dreaming of discoveries. And then it wasn't until her college internship in clinical microbiology that she really got fascinated by the microbial world and in particular microbial evolution. So when she learned about plasmids and horizontal gene transfer, she thought, aha, this is a tool for me to study microbial evolution. She points out that this project started on just an ordinary day in the lab. Her PI, Hero, just said, what do you think carries more DNA? A small multi-copy plasmid or a large low-copy plasmid? So she gave her answer, probably the small multi-copy one. But then she's like, wait a minute, let me take a look. So her first look at a data set surprised her that almost all the plasmids she looked at seemed to have about the same amount of total DNA, regardless of their size or copy number. So she immediately called Hero, come look at these results. And that was the birth of this project. She did say it was a lot of work. I mentioned that sometimes she and her teammates had 6 or 7 computers running simultaneously. But she is really grateful to her PI and also the lab mates who helped inspire, especially the second author, Nacho, who was incredibly helpful, especially since they were both still learning the field. I already mentioned what a thrill it was for her to go to that conference in Hamamatsu, Japan, and meet another team that was working on similar questions that Mark described. She does have some advice for aspiring scientists. She says, don't be discouraged by how slow things can be. Sometimes small steps lead to big questions. Always ask for help. Don't underestimate your ability to learn something new, even coding. Listen to people around you, learn from them, and realize that failure is part of the process. It's why we call it research and not search, as I like to say. And never let go of your curiosity. So when she's not in the lab and running multiple computers to process data, she loves coming home and telling her boyfriend everything she'd done that day, whether he understands it or not. And slowly but surely, he's learning. She also enjoys spending time with friends and family to recharge and disconnect from research, and also enjoying hiking in the mountains or walking through a new city or reading a book. Great ways to kind of clear her mind so that when she comes back to the lab, she's ready to go. So thank you. Yeah, thank you for sharing that, Paula. And also I want to thank Hero for taking the time out of his parental leave. He and his wife have just welcomed a new baby. And despite that, he was able to respond to my email and direct my query to Paula. So thank you both and congratulations on this beautiful work and your lives.
So Mark, I'm sure this question has been asked and answered. Do plasmids confer a fitness cost on bacteria?
Yeah, I absolutely can see it in my own lab when I do, for example, serial passages of anything carrying a plasmid, and they tend to lose the plasmids quickly.
Yeah.
And those are based on plasmids with high copy number. The low copy number plasmids, on the other hand, seem to have this stick-to-itiveness. And then I wonder about the ones that are putting metabolic genes from the chromosome onto them. As I mentioned with thiamine biosynthesis with one of the megaplasmids rhizobium has. So I think that you have to ask what conditions you're using and whether that fits and I'm going to use this term incorrectly, the ecophysiology of the situation.
In the lab, we always used to have an antibiotic selection marker, right, to make sure they stayed.
Right.
But in nature, that's not always the case, right?
Mm-mm. No, I mean, I know a few things that will hold on to a plasmid under stress. But if you put part of the metabolic pathway onto it, they probably would. It's a great question. Yeah. I think Rich Lenski's looked at fitness costs of small plasmids. But again, Vincent, how do you study that? You basically do serial transfers.
Mm-hmm. Well, with and without a plasmid.
Mm-hmm. Or selection pressure.
Yeah. So I like to think of there's this constant talk between the chromosome, the plasmids, and even DNA that comes in, endogenotes that come from outside the cell. And I think the question is what becomes fixed.
Talk is such a nice way to phrase it. They're competing for resources. They're trying to make two of themselves. That's it.
Oh, yeah.
When a bacterium is in a environment full of antimicrobial compounds, then acquiring a plasmid with the resistance genes restores the fitness, right?
Mm-hmm. Yeah.
So it can go either way.
And if you look at organisms that are naturally competent, like Acinetobacter, it is very clear that one of those strands is chewed up for food. And the other one could potentially be incorporated if there's some benefit for doing so, which means it happens. And then the question is whether there's a selective benefit of doing so.
Are all, by definition, plasmid is a double-stranded DNA circle. Is that correct?
So far, you notice I answered that carefully. I do not know of any single.
You mentioned it, the Streptomyces plasmids.
No. Linear is not the same as single-stranded.
Okay.
And I'm trying to, I was thinking about how M13
M13
replicates and does a rolling circle and has a single strand, but that's not a plasmid.
That's a phage.
Yeah. And there are some phages. Well, there you go. The edges
we used
blur.
to take the M13 genome, double-stranded DNA circle,
right?
Put genes into it.
Yep.
And then put that into a cell, and then the phage would come out with single-stranded DNA, and then you could sequence it very quickly, right?
I remember that.
Remember Joaquin Messing over at Rutgers?
I spent a lot of hours doing that. A lot of hours.
Yeah, tons. But the thing is, when you put the double-stranded DNA, it's almost like a plasmid. You transfect it into cells, and it reproduces as a autonomous double-stranded circle, so kind of plasmid-like, right?
Yeah, and that's why I say these things are kind of blurred lines.
Well, they're terms we come up with to communicate with one another.
We make up the terminology. What do we know, right? We can't talk to them. Anyway, Mark, thank you so much. That was very interesting.
Yeah.
I like plasmids.
Isn't it great that they both found the same relationship using two different techniques?
Yeah, biology doesn't lie, right?
Right.
It's the humans that lie.
Yes.
And on that sour note.
Ooh.
None of us here on this program, but other humans are known to lie. That is episode 337 of TWiM. You can find the show notes at microbe.tv/TWiM. If you enjoy these programs where we give you science, a bunch of scientists talking about science, it's a lot of fun for us. We hope you like it. We'd love to have your support. We don't take ads. We only take contributions from our listeners. microbe.tv/contribute. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Our guest today, TWiM adjacent, Mark Martin from the University of Puget Sound in Tacoma, Washington. Thank you, Mark.
I'm always happy to be here.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.